So I looked at who does what, and I basically found my current supervisor, Dr. Amra Zayed, and he happened to work with honeybees, and I liked his work, so I interviewed with him, and I've never worked with honeybees before I joined my master's program. And thank God that I love them. Because otherwise, it would have been torture, but luckily I fell in love with them. They're interesting. They have lots of behaviors. They're kind of really smart for an insect. And you get to be outdoors and enjoy the weather for once and not just sit in the computer, like in front of a computer all day.
Okay, great. So you know what, without further questions, why don't we just get started?
Okay, so thank you all for tuning in. So I'll be talking about my PhD work of neonicotinoids and honeybees. So first, a little bit about neonicotinoids. So they're a systemic pesticide, which means that, for example, in corn, they're applied on the seed, and then as the seed grows, the pesticide grows into everything. It goes into the roots, into the stems, into the leaves. So one advantage of that is that you don't have to spray it later on. And of course, that this way, all of parts of the plants are protected. It is, as the years went on, you can see on the graph that it became more and more commonly used. So all of these names are different types of neonicotinoids. And you see here the graph until 2011, and the amount of neonicotinoids we were using throughout the world. And now it is the most commonly used insecticide in the world, kind of period. As people started using it more and more, we had problems in Canada where beekeepers started phoning in and saying that their entire colonies were dead in the middle of spring, which is highly unusual. And you can see these red dots is where those incidents came in. So it's mainly Southern Ontario, a little bit of Quebec. And it just so happens that these are the areas where we grow corn. So the second figure is with the red dots is where we actually grow corn, and you can see kind of a substantial overlap there. So that's what kind of made us focus our experiment on corn. So before our experiment, there were some studies that showed that neonicotinoids do have negative effects on honeybees. So these are studies were done in labs. So you take a bunch of bees, you put them, in a box, you feed them some syrup with some neonicotinoids, and then you see what happens to the bee. Now, a lot of those studies, though, were criticized for not being field realistic. So first of all, you take an isolated bee, you kind of force feed her. She's isolated, she's not in the colony. And of course, also the question was, how much of the pesticide do you feed her? Because of course, the dose makes the poison. So it was criticized. So a lot of people dismissed this as kind of, this is not field realistic, so we shouldn't really pay attention to this. At the same time, there were studies that were done in the field, and a lot of those studies actually failed to detect any impacts on the colonies, on mortality, on winter loss. But the question actually arises, are these studies field realistic? And a lot of them were done on canola. And as we saw from the previous picture, the problem areas are not near canola, they're near corn. So this is kind of the question is, well, what is field realistic? So this is what we focused on the first part of our study, to actually find out what's going on in the field. So in order to do that, we took a bunch of honeybee colonies, and half of them we placed far away from corn, and half of those we placed right next to cornfields. And then what we did is we sampled them for pesticides, and we also observed several behaviors to kind of see how they're doing. We sampled freshly collected nectar, we sampled freshly collected pollen, nurse bees, forager bees, larvae, and freshly dead bees. And we also sampled them from the beginning of May till the end of September, which is the beekeeping season here in Southern Ontario. So this way we kind of wanted to really get a complete picture of what's going on. Then we send all of those samples, we sent for analysis, and we actually tested for 231 different chemicals, because maybe it's not neonicotinoids, maybe it's something else. We really need to make sure we know what is out there. And in fact, we did detect a lot of chemicals. We detected 26 different chemicals. So this is what those chemicals were. So a lot of miticides, fungicides, neonicotinoids, herbicides, and a little bit of non-neonicotinoid insecticides. So just as a side note, the miticide here is actually a single miticide. It's Coumaphos. So Coumaphos used to be used to control the Varroa Mite. The Varroa Mite is a parasite on honeybees, and it's really causing a lot of damage. But it's no longer used, mainly because the mites have developed a resistance to it, so it doesn't work anymore. It's also not great for the bees. But the reason we're still detecting it is because it's fat soluble. So it sticks in the wax and actually in the bees themselves for years and years on end. So that's why we're still detecting it, even though neither of our beekeepers have used it for years. Okay, so we see there's a lot of different chemicals inside of our colonies. So why am I picking on neonicotinoids? How do I know it's neonicotinoids and not something else that's causing all of the problems? So one way to compare these chemicals who have very different kind of properties is to assess the risk. So we take the actual amount we found in the field, and we divide it by the LD50. The LD50 is the measure of toxicity of the chemical. So it's how much it takes of this chemical to kill 50% of our test animals. So this way we can compare different chemicals with different properties in kind of a meaningful way. And what we found is that neonicotinoids are by far the thing that has the highest risk to bee health. So that's why we focused our analysis on neonicotinoids from now on. So next question was, where did we find those neonicotinoids? Mainly in pollen. Pollen was by far the thing that had the most pesticides in it in general, and it's the thing that had the most neonicotinoids in it. The second largest group, which is interesting, is actually freshly dead bees. So we did find a lot of neonicotinoids in dead bees. I also want to point out that we even found some neonicotinoids in larvae, which means that the foragers bring in pollen, then the pollen gets stacked, the nurse bees eat that pollen, they digest it inside their head with a special gland, and then they spit out the food to the larva, and neonicotinoids manage to survive this entire process. So this is a little concerning. So pollen was the biggest thing that we found. So the next natural question was, well, which plants? Because pollen is comes from all over. So which plants are contaminated with these neonicotinoids? So we did a microscopy, which you take the pollen and you just look at it under a microscope, and by the shape you can see which plants it came from. And what we found is that the most common pollen that we found in our neonicotinoid samples was willow, clover, and buckthorn. So all of these plants are non-target plants. So they're wild plants that are grown that are not sprayed or not used with neonicotinoids. So this is quite a bit concerning. We didn't find it in corn pollen or soy pollen in our samples, really. It was really willow and clover and buckthorn. So that's a little alarming. Now, the second question is, well, how did neonicotinoids get to all those plants? We don't use neonicotinoids with these plants. They're kind of grown on the margin of our fields. Well, actually, there were studies about this. So this is a study that was previously published in the journal Nature. And you can see here is kind of the how and where neonicotinoids go after the crop is planted. And you can see that most of the neonicotinoids actually end up in the soil and in the water. And if you have lots of neonicotinoids with water, well, willow, for example, is a tree that has, it's very water intensive, takes up a lot of water. So maybe that's why we find a lot of neonicotinoids in it. So next is when. So here I'm going to show you a graph. So A is the exposed sites and B is the unexposed sites. Again, early May to late September. And the little triangle is when corn was planted. And the darker the blue color is, the more neonicotinoids there are. And this is what we found. So first is that we found neonicotinoids inside the bee colony for 3 months on average in the exposed sites. Nobody expected us. We didn't expect this. You know, people thought that neonicotinoids were this kind of one-time hit during corn plantation from the dust, and that's it. But in fact, for 3 months on average, we found neonicotinoids in the colonies. In fact, in 2 of our sites, we found neonicotinoids before corn plantation, which indicates these are neonics that were there from the previous year. So there's probably an accumulation effect in the environment. Another note is that we even found neonicotinoids in our sites that were far away from corn. So of course, for a much shorter time and much lower dose, and not all of them, but they were still present in certain of our sites, and we think that's because of runoff. So some of our sites were downstream next to a creek from fields. And of course, planting by municipalities of plants that were treated for neonicotinoids. Okay, so this is, we did kind of an overview work to try and see what's going on in the field, but now we need to know, okay, so this is happening, what kind of effects does it have on the honeybee colony? And the best way to do this is to do experimental work. So this is exactly what we did. Is we took a bunch of colonies in our apiary. Half of those colonies, we fed them clothianidin, which is the most common neonicotinoid that we found, and half of them we didn't. And we also fed them for the duration and the kind of dosage that we found in the field. So 3 months, we have a little bit higher dosage in the beginning, and as the 3 months go on, the dosage trailed off to lower and lower. And then we monitored the behavior of our bees for those 3 months, and we for every 2 weeks we looked at our bees. So the first thing that we did is we looked at hygiene. So hygiene is very important for honeybee colonies because there's a lot of bees. As soon as a bee gets sick, it's really important to get her out of the colonies so she doesn't start a pandemic. So one way we test this is we take a brood, which is basically baby bees that are sealed underneath the wax, and they're growing to become full adult bees, and we kill a bunch of them with liquid nitrogen. So this is kind of instantaneous death. They freeze, and then we put this frame back into the colony, and we come back 24 hours later, and we see how many of those dead pupa the bees threw out. So in 24 hours, we get a measure of how clean the bees are. So first in 2014, which is the field study, we noticed that towards the end, by September, the colonies that were near corn were much less hygienic than the colonies near corn. The colonies near corn were much less hygienic than the colonies far away from corn. And in our experimental work, we also saw that as time went on, the exposed colonies became less and less and less hygienic. So next, we looked at the queen and the presence of the queen in our experimental work. And we saw that, again, as time went on, the exposed colonies had real trouble replacing their laying queen. So sometimes honeybees will lose their queen, she'll either die or they'll swarm. And here it's really important for the colony to quickly build a new queen. If they fail to do that within with the available eggs, so unless there's human interference, the colony will die because only the queen can lay eggs that can become future queens. So if you miss that window, you have no more eggs. That's it. There's no more queen. Nobody can lay eggs anymore. And that's it. The colony will dwindle and die because there's no new workers being born. So in our exposed colonies, they really had trouble replacing their queen. Another fun thing that we did was we put little microchips on bees. So we took sealed brood, so it's bees that are not born yet. We took them into the lab. We put them in an incubator, and then as the bees were emerging, put little microchips on their backs. And then we took all of those bees and we placed them in the third unrelated colonies. And that colony had a sensor on its entrance. So as the bee went out, that sensor read the microchip, and as the bee came back, the sensor read the microchip. And each chip had a unique identification. So this way, I can track the flight behavior of each individual bee throughout her lifetime, which is really, really fun technology. So what we found with this technology is that, so first of all, bees have something called orientation flights. So this is when a bee just becomes a forager. She starts flying outside the hive. She takes these short flights where she just flies around her own colony to try and remember where it is. So that later when she takes a foraging flight, she comes back and she's able to find her colony. And what we found is that the exposed bees, in the beginning of their foraging experience, took a lot of more of these flights that are control bees. During the foraging flights themselves, as the bees actually grew older, the exposed bees took longer and longer and longer flights. Now, you can also see that the red line is a lot shorter than the blue line. And that's because when we look at the life expectancy of our bees, we notice that the exposed bees live a lot shorter lives than our control bees. But it's 23% shorter. It's quite a significant amount. Now, another thing I want to kind of reiterate here is the fact that all of these flights and the life expectancies, the bees were exposed to neonicotinoids only in the original colony as larvae. They were not exposed to neonicotinoids as adults. Both the control and the exposed bees as adults were in the same colony that was not treated with anything. So this means that all of these kind of changes, they're probably kind of developmental changes that happen when the bee is still a larva. Okay, but that's not the end of the story, because we found a lot of different chemicals in our colonies. So as we might remember, when you mix different chemicals together, a lot of different things can happen. They can make things worse, they can make things better, they kind of cancel each other out. So we wanted to see what's happening with this mixture of chemicals that we have in the field. So what we did is we tested two different neonicotinoids, clothianidin and thiamethoxam. So these were the two most common neonicotinoids that we found in the field. And we tested them also together with lineron, which is a herbicide, and a boscalid, which is a fungicide. And on the y-axis, you can see the LD50. So again, this is the amount of chemical it takes to kill 50% of your test animals. So actually, the lower the number, the more toxic the mixture is. So by themselves, just the neonicotinoid alone, they kind of had the standard LD50 values. Together with the herbicide, there was no statistical difference to the mortality or the toxicity of the mixture. But with the fungicide, the toxicity of the mixture really, really increased. So it's doubly as toxic as just the neonicotinoid alone. Now, the amount of boscalid that we added was very low. And by itself, when we just feed this amount of boscalid to the bees, it kills 0% of the bees. So it's not toxic at all at this amount. But this amount together with the neonicotinoid and the mixture becomes doubly as toxic as just the neonicotinoid alone. This becomes especially concerning when you learn that the seed coating for neonicotinoids is patented with 3 different fungicides. Okay, so what is field realistic? We learned that field realistic is exposure through pollen. It's exposure for average of 3 months. And it's exposure to a variety of pesticides at the same time. We also learned that realistic exposure leads to reduced hygiene, reduced queen replacement, reduced life expectancy, and abnormal flights. So I'd like to thank you for listening. I'd like to thank the major funding of this project, which was UMAFRA. I'd like to thank my personal funding, which was OGS and York University, Université of Laval, which did the Quebec side of this project. Of course, my amazing field team that spent countless hours with me in the blazing sun. And again, thank you for listening.
Well, thank you, Nadia. That's I got to hear your talk when you spoke in London, which is where I met you at the. It was at the Ontario Beekeepers Association.
Oh, hold on, sorry.
So those of you that are listening, please feel free either if you want to type in some questions that you may have for Nadia, or you can unmute yourselves as well. There's not too many of us, so please feel free to ask some questions. So yeah, there's a lot of information there. I mean, so it shows you that it's quite serious. We need to be paying attention. And so I have a few questions before maybe everybody else gets started with all their questions. So, I mean, I would imagine that for native bees that are also living in those fields, I mean, it must have quite an impact on them as well, and plus all the other insects that are there.
Right, so that's kind of, we of course focused on our honeybees just because it's simpler, to be fair, the colonies are right there. But it is very disconcerting for native bees. Especially certain bumblebees that come up early in the spring. They're a single bumblebee that will start her nest, and the only thing she has to eat is willow. It's the only thing that blooms early in the spring. And as we saw, a lot of the willow has neonicotinoids in it. So if that single bee dies, that's it. There is no colony. She can't start her colony. She can't lay her eggs. If a single honeybee worker dies, I mean, there's 10,000 more from that colony where she came from. But if a bumblebee queen dies, that's it. There's no colony, there's no kind of next generation. So it is very concerning. Of course, there's also the issue that, we use honeybees for the official kind of PMRA testing to approve new pesticides and as a stand-in for all bees. And that's not the best system, because honeybees are actually kind of very unique because they're a social bee. And in fact, most of our native bees, 99% of them are solitary bees. They live on their own. A lot of them are ground nesting bees, which means they dig holes in the ground, which is where all the runoff water with these neonicotinoids ends up. So yeah, it's very concerning. It's probably worse for our native bees and native insects than for the honeybees.
Right, and we're not, there's not as much research, I guess, for the native bees, so knowing exactly what the impact is for them as well. So let me ask you another question. I mean, it was obviously quite surprising to see how the pesticide ends up on other plants, on the pollen. And do you think it's impacting our cities as well? Was there any research done in cities to indicate that the neonics are in the pollen here in the city?
So a couple of our sites were in Toronto for our unexposed sites that were supposed to be far away from corn and without neonicotinoids. And in fact, in one of those sites, we did find neonicotinoids. That was an in-city site. So it's getting here somewhere. There were a few studies that were done that looked at mainly the plants that were being sold at places like Home Depot, that did find that those plants are treated with neonicotinoids, and there's neonicotinoids in the pollen. So of course it's very concerning because, for example, if you want to start a pollinator garden, you go out there, you buy these great plants, you think you're doing good, and then it ends up they're all poisoned. Kind of this is where the consumer voted, and there was actually a big kind of uproar about it, and Home Depot actually stopped carrying certain plants that are treated with neonicotinoids. And nowadays, if you go and you buy plants, you can look for a sticker that says not treated with neonicotinoids.
Right. So I mean, I have more questions, but maybe I'd like to open it up. Would anyone they could please unmute yourself and ask a question or type it in. Oh, sorry, I think I see some typing here. Can you comment? So this is from Lauren Whitmer. Can you comment on how much neonic exposure there is to honeybees in urban environments compared to rural?
So from what we saw, a lot less. So again, a lot of the unexposed sites, just by the nature of the geography of Southern Ontario, if you want to be, 3 kilometers away from a cornfield, you end up in the city. So you get a lot less exposure for a shorter amount of time. And in certain sites we had inside the city, we had no exposure to neonicotinoids.
Okay, thank you. So Susan Blaney has a question too. Has there been any response by the chemical companies to your research?
So there was a little bit of response, mainly ignored, which I guess is also a response. I think we found one person that was tried to kind of show all the things that we did wrong in our study. Unfortunately, a lot of the criticisms he had, we actually addressed. So kind of if he read it closer, he would have seen that we actually addressed a lot of the issues he brings up. Other than that, no. But to that effect, we actually did have an impact. The United Kingdom banned neonicotinoids about a year or a year and a half ago. And in their official report, they actually cited all of the scientific information that they used, and they actually did cite our study in their decision to ban neonicotinoids.
Congratulations.
Thank you.
That must feel good, doesn't it?
It does, that somebody's paying attention.
Yeah.
But to that effect, the government, I believe it's Canada or Ontario, I'm not sure, but they actually decided not to ban neonicotinoids. And in their report, they actually didn't cite anybody. And in fact, they concluded that it's not a problem for non-flowering plants. And of course, this is exactly counter to what we showed. So corn is not is pollinated by wind. It's not attractive to pollinators. So by their logic, well, then it's not a problem for the bees. But this is exactly what we showed, is that next to corn, we actually have the problem.
Yeah. Okay, I have another question here from Dinah Robinson. She says, what are your thoughts on the federal government plan to phase out just 2 of 5 neonics by 2021?
Yeah, so it's kind of odd because so imidacloprid was kind of the first big neonicotinoid that was used. So a lot of the research was done on imidacloprid in the early days. And it is very toxic. It's actually a lot toxic than some other neonicotinoids. And of course, when it degrades, it was found that kind of the degraded form is just as toxic. So when we talk about like, oh, after, it just degrades and that's it, it goes away. Well, that's not true. When it degrades, it becomes a different chemical. And we need to test that chemical. And in fact, one of our most commonly used neonicotinoids today, which is now clothianidin, degrades into a different type of neonicotinoid, thiomethixane. So just because it degrades, it doesn't mean it becomes inert or goes away.
Right.
And just because we're phasing out 2 types of neonicotinoids, they're gladly use different types of neonicotinoids. And there's a reason these pesticides are in the same group. They have the same mode of action. They have the basic same chemical structure. So there's no reason to think that one will be better than the other.
Okay, and I guess there's a part 2 to her question. She says, also wondering what you think about cities that issue permits for pesticide spraying because they only have a ban on cosmetic application. That's the situation here in Ottawa. Do you have examples from other cities? So
here in Toronto, we have bans, so regular people can no longer buy pesticide to spray on the lawns, but golf courses can. And golf courses are actually one of the worst offenders for pesticide use and abuse. Because, you know, everybody goes to a golf club, it's super expensive. You want every blade of grass to be perfect. The only way you get to do that is if you drench everything in pesticides. It maybe reduces use. It does reduce use from the common people, right? Like for regular people. What it does also do is gets people used to non-manicured lawns. Maybe you instead, you just plant some flowers. If you're tired of your dandelions, you uproot your lawn and you actually plant some flowers. So in that sense, it's actually kind of good. And of course, I've seen this change lately, which is actually a really good change for next to road and next to dividers and next to highways, you get these plots of land. A lot of times there were manicured lawns for some reason. And nowadays, more and more municipalities let them just grow because it's a waste of money to manicure them for, nobody cares. You just drive by it, and then, you have to put pesticides on it, and why would you?
Right, thanks. So, Dinah says, great comments. Thank you very much for the additional details. And there's another question from Lauren. He wants to know, how much impact do neonic treated seeds, example, annual flowers, planted in urban gardens have on honeybees?
So that's, I don't know. That's the short answer, because we did kind of focus on the field studies on the corn, because that's where most of the commercial beekeepers keep their bees. For city, I guess the short answer is I don't know.
Okay. Okay, so I think I have some more questions. So Bee City Canada is all about action. We're trying to get the cities, what they're planting, reduce their pesticide use. So what do you think we could do? What do you think individuals can do? Like, how do we get our government to start banning these pesticides?
So I'll say the most common and kind of cliche thing is at this point, write to your MPP.
Okay.
Now, I know it's a little kind of cliche, nobody wants to do it, but actually from what I understand, if you write to them, especially if you write to them on a physical piece of paper and send it, they actually read those letters, because it means the person was engaged enough to, go out, buy a stamp, find out their address, which means that if there's enough people doing that, there's probably a really strong demand for it in their constituents. On kind of a more kind of local level, what you can do is you can pressure your municipal government. So municipal government is a little easier because as a single person, you represent a much larger percentage of their voting base than from, provincial standing point. And maybe you can pressure them to make sure why the golf courses get an exemption for pesticide use.
Right.
Why does their lawn have to be manicured but mine can't? Kind of seems unfair. We either decided that the pesticides are harmful or they're not. It's not like they're not harmful at a golf course, but they're harmful on my lawn. And of course, you can start, there's these great things here in Toronto, the community garden initiatives, more and more people starting to put kind of bee hotels, that's for native bees, in their backyard, or just keeping a few honeybee colonies in their backyard. And of course, planting native bee-friendly flowers.
Right. Yeah, thanks. I wrote those down, and we'll make sure that our representatives from our bee cities know about this, because I think education is another important aspect too. People just don't know. I think we tend to think that our governments are taking care of us, and that they're doing studies on all these pesticides, and they're going to make sure it's safe. So how is it that Europe? Do you know what's going on in Europe? Could you share with us what's going on?
So from what I remember, so UK outright banned it. The EU had a partial ban for quite a while, but the partial ban was only in certain types of crops and not others. And I believe in, I think since then it kind of become a more local issue. So for example, a few years back when the EU had just the partial ban, I know a locale in France, a province in France, they outright banned it. So just because it's allowed federally doesn't mean it's allowed provincially.
Okay. So do you know of any other provinces? Like I know Ontario, in the last government, they put together the Pollinator Action Plan, and Lauren was involved with that, where they had a partial ban or a reduction, I guess. Do you know about how that's working with this current government, the new government we have?
So I think I haven't heard anything, so I think they just left it as is to kind of roll the plan as it was. That said, the plan had a lot of holes in it. So, for example, the plan was to reduce the amount of pesticides used, and one of the ways is for farmers to have to prove they need the pesticide. And of course, the way they do it is there's an inspection that comes by, and the farmer shows, here I have this particular pest that this neonicotinoids target. But then I also heard that kind of people sell those pests in the jars. So when the inspection comes, you can just show the inspection, here, I got this pest, I have to use it.
Right. Oh, that's a good point. Yes.
There's a lot of holes in the system.
Right. Yeah. So, so I've got a question here from Susan Blaney. She says, Friends of the Earth tested bedded plants in Southern Ontario for pesticides and found banned pesticide residues. I think a consumer campaign to demand the nurseries clean up their act would be a good direction for us to take. I guess it's just a comment.
So yes, actually, it's also the question where those plants come from. So a lot of kind of plants that are sold as like bouquets actually don't come from Ontario. A lot of them come from, I think, I want to say Kenya is a large producer of a lot of those plants, but I'm not sure.
Maybe Colombia too, or South.
Yeah, so they come up from abroad. So just because they're banned in Canada, again, they're not necessarily banned there. Right. So one of the things we have to demand is perhaps a label to see, okay, what was this treated with?
Right.
So when I get to choose between, this type of plant or that type of plant at the store, I can see, well, this one wasn't treated with neonicotinoids, so maybe I'll buy this one. The nurseries in Ontario, from the last thing I've heard, they actually do use IPM quite a bit. So this is integrated pest management. So they used a lot of different options before they go to chemical pesticides.
Right. Yeah, I mean, I think people maybe people that are on this talk listening to you maybe know, but I mean, there's the Neonics, but there's lots of other pesticides as well that are out there. And so how, like, for example, like I think you mentioned there were 25 other different kinds of chemicals, or how many toxins are these poor bees subjected to in their life?
A lot, right? And that was kind of part of the point why we tested the mixtures as well. Because of course, a chemical cocktail, it doesn't always have the same effect as just a single chemical. Yeah. And that is a problem, because a lot of the times when a new pesticide is introduced, it's only tested in its pure form, just the chemical itself. But when that product is being sold to the farmers, it's being sold with a bunch of other stuff.
Right.
So for example, the neonicotinoid seeds, they're of course sold with the neonicotinoid, but they're also sold with a bunch of fungicide. So because, you have bags of seeds in storage, you have to make sure that they don't get eaten up by the fungicide while they're in storage or while they're, before they grow. But nobody tests the actual mixture that's being sold to the farmers.
Right. Yeah, good point. And from my understanding as well, there are a few more questions which I'll get to. From my understanding as well, though, that the bee itself has a pretty low immune system, and so it needs to forage to forests even, for to looking for fungus for it to intake is that true? Is that?
So there is some evidence that certain type of fungi, when they grow inside the colony or they eat them, kind of helps boost the immune system. It's a very particular type of fungus. But one of the ways we actually think that the fungus affects our mixtures, the one we tested, because we tested this on just singular bees, not in the colony, is the way these chemicals act. So the fungicide, the one we tested, the way it acts, it actually blocks the system that detoxifies chemicals. So if it blocks that system in a honeybee, and she's not exposed to anything, then she's fine. But if blocks the system and she's exposed to chemicals that she can't process them, and she dies.
Right. And I think what your research showed too, it affects so many different behaviors of the bee. It's not just one. How far they forage, they're not as clean. And of course, the cleanliness of the hive is extremely important, correct?
Yes, absolutely. Right. You have 20, 30, 50,000 different individuals living all together in a very, very tight spot. So it's very easy for disease to spread around. Sorry. So, yes. Sorry, what was the question?
Just about cleanliness of the hive.
Of the hive, yeah. So they kind of, bees are actually really, really clean. They only poop outside, even during winter when they're months on end inside the colony. They actually wait for warm days to go outside and poop. They don't go inside. They clean their hive constantly. They constantly take out dead and sick bees. They kind of throw them out because they don't want to start a pandemic. Social immunity is super, super important for these bees. Absolutely.
Okay, so here's some questions. So Julianne Lebrecht, she's from Ottawa. She says, I'm wondering if there is any scientific connection between neonics and the health of other animals or humans.
There's actually quite a bit of work on birds, for example. One of our York University professor, Bridget Stachbury, they actually published a book, Silence of the Songbirds, that shows, so all of our research, it shows that a lot of the songbirds, they tend to be smaller in body size. They are declining, they're getting poisoned by neonicotinoids because a lot of them eat insects that have neonicotinoids or seeds that have neonicotinoids in them. So that's definitely getting, so birds are definitely a big hitter. There's evidence on aquatic life because, again, neonicotinoids are water soluble, so they end up in a lot of watersheds and a lot of rivers. So there's some evidence that it negatively impacts the kind of the frogs, but also things like water striders. At the end of the day, they're insects. Right. Dragonflies, these are insects that in their larval stages, they live inside the water. And neonicotinoids are insecticides. They're designed to kill insects. And on humans, there's a lot less literature because, of course, it's hard. We can't really do experiments on humans. There's been some evidence of negative impact from Japan. A few years back, there was a big case where a lot of people got super sick in the town after application of neonicotinoids nearby. This is kind of correlational evidence, of course. But there's now more and more evidence on mammals, on rats mainly, that show that exposure, especially during pregnancies and these kind of fetal stages during kind of very vulnerable times in development, has negative effects.
Okay, so Teresa Atkinson has a question here. She says, sorry, I don't have a speaker. I noticed at Home Depot that rather than starting a particular plant, a citrus plant, rather than stating a particular plant, a citrus tree was neonicotinoid free. It stated that it was treated with imidacloprid. I'm not sure if I'm pronouncing it correctly. I don't think that most people in the general public know the various names of neonicotinoids. Is there any kind of published information that is accessible to the non-scientific general public?
That's a great question. So imidacloprid is a neonicotinoid. So
Yeah.
I don't know what the logic is there. There is, there are a few actually Toronto initiatives where I think initiatives from Guelph, Pollinator initiatives that actually publish little booklets about kind of the types of pesticides or community gardens, types of plants to plant. I wish I had it handy. I don't. Perhaps later I can send you an email with links to it, and you can send it to everybody.
Yeah, exactly. We'll do that. We'll send that with the recording as well. Any information that you can share. So if anyone has looking for information, that would be, we can see if you have some. You know, and maybe we could write I'm just thinking about what you said, what we could all do is write a letter, and maybe we could have you know, kind of a letter that everyone could write themselves and mail it off. Maybe we could send
like a template.
Yeah, I think there's some bee stamps too out there. Not to forget. I mean, everyone knows how important pollinators are, and you know, insects are dying. I mean, these studies that have come out are pretty alarming. What's going on around the world? So, you know, what's it going to take? I don't know. We were all asking each other. Maybe we could all help each other and what we could do. So, Teresa has another question. She says, "Are there any studies done on mason bees?"
So I know one researcher who studies mason bees, but I don't think she's done any pesticide work. Mm-hmm. There's a researcher in, I want to say Brock University, that does a lot of work with mason bees, but I don't think she's done any work on pesticides in particular. Right. For the native bees, there's some bumblebee work, a few on the leaf cutter bees, and again, mainly because these bees are used in agriculture. Right,
right. So I did have a question. Yeah, so, you know, there's always seems to be something new on the horizon from some of the chemical companies of what they're going to put out next. You know, the next generation of pesticides, herbicides, fungicides, all this stuff. So do you know what they're planning to launch?
Yeah, so unfortunately I don't remember the name of it because, again, I didn't study it. There are some researchers on the edge of that who are actually very concerned. There are types of... there are different type of pesticides, so they're based on a different chemical structure. Now that said, so kind of we will need pesticides if we keep growing, doing agriculture the way we do. We have to have pesticides. There's just no way to grow food without them in kind of the manner that we do. But that being said, we also have to push on these companies to be better. I mean, the first pesticides, kind of the big one, was DDT. It was disastrous. It made many people sick. It killed many wildlife, and people pushed back. And the new pesticides are not as bad as that one. So I think if we keep pushing and pushing back, they'll have to come up with better and better solutions.
Mm-hmm. And a question too about farmers, because it seems that, you know, the main the main groups of people that are purchasing this are farmers. So what can we do, or what could Bee City do, you think, to maybe help support farmers so they could look at different alternatives to use less pesticides, or to have different ways to grow their crops, or maybe the end user about what we're buying? Are we encouraging the use of pesticides?
So here corn is one of the big issues, and it is the thing that we use neonicotinoids on a lot. I think part of the reason people grow a lot of corn is it's partially subsidized by the government. And that's why you see corn syrup in everything, for example. In every processed food, there's corn syrup in it because it's a cheap sweetener, because it's subsidized by the government. I mean, honestly, I would cancel all subsidies for agriculture, but the problem with that is that a lot of farmers would go out of business. But then again, if you're growing corn, that the only way it's viable on the market is for the government to pay for, half of it, maybe you shouldn't be growing corn. But that's kind of a big picture one. I don't know if it's ever going to happen. On the more local scale, actually a lot of farmers do care about bees, and a lot of farmers do care about wildlife. And I know of stories that in 2014, for example, a few Quebec farmers, they physically couldn't buy seeds that were not neonicotinoid treated. It was just everything that was available, and that's it, and they couldn't buy it. Now there's more pushback, so now there's available seeds on the market without neonicotinoids. A lot of farmers believe it's just the cheaper alternative because although the seeds cost a little more, you don't have to spray them, you get less damage to your crops. But now new studies come out that show that it's not always the case, that in some cases you are losing money. And at the end of the day, we do need to support the local farmers. If you have a farmer's market that's growing not corn, so cauliflower, broccoli, whatever it is, apples, cucumbers. I know we have a very short limited growing season in Canada. So, I mean, you can support them because these types of crops, they don't use neonicotinoids. They're kind of better for the environment because they tend to be on smaller plots of land. So things like that.
No, thanks. Those are great suggestions. So David Mesfeld from Calgary says there's a young farmer in Saskatchewan who only uses pesticides for the first 2 years. And after that, he is totally pesticide free. So I imagine there are farmers that, would like to change, and maybe the government could help these farmers to grow food a little more differently. I mean, there's regenerative agriculture, which is something which is gaining momentum, especially I think in Europe and other parts of the world, a little slower here in Canada and North America. Are you familiar with that, or can you?
So it's kind of funny because part of the reason our kind of Canadian and American agriculture is actually a little behind in its technology. And part of the reason is land here is cheap relative to Europe. So, for example, Amsterdam actually has one of the most advanced agricultural use of agricultural technology. And that enables them to practically use kind of very little pesticides. And, they use crazy things like drones to pinpoint things, to, there's a new kind of AI technology for greenhouses that lets a little robot like pluck weeds instead of spraying it, or instead of going and doing it by hand, like crazy things. Cool. But here in Canada and US, land is cheap. So growing this vast kind of land of corn and, wheat, canola, whatever else, and even if you just have to use pesticides, and even if you lose, 10, 20% of your crop, just because the size, it's still economically viable. And of course, technological investments, especially early adopters, is very expensive. So maybe the government can help out to those early adopters, that because new technology is always very expensive. So maybe they can help these new adopters if they choose to use some of these technological innovations instead of using pesticides.
Yeah, that's good. That'd be really cool. Maybe we could actually have some of that on our website as well. What other countries are doing farming. I like that new technology, a little drone going out and seeing where there's a problem, and then
it's amazing.
Just picking up the weed. I love that. Let's see if there are any other questions or anything. I think he says, I think he's got 40,000 acres in Saskatchewan. Wow. And he's pesticide free.
That's amazing, right? That's great if it's not using pesticides. Of course, what's kind of interesting, even the conventional farmers, they're supposed to cycle their crops, they're supposed to cycle their pesticide use.
Yeah.
because you do get resistance to these chemicals. But some of them don't, and some of them, even if they do follow the correct recommendations, a lot of those recommendations are made by the pesticide companies themselves, so their efficacy is kind of not always known. So yeah.
A lot more things that we can all do. So are there anyone else, any questions? I think maybe that's all. I can't thank you enough. It was a fantastic presentation, and just so much knowledge that you have that you'd shared, and then solutions of what we can do. And if you think of other solutions, please pass them on to us, and we'll see what we can do in our brochures as well. We tell people what to do is certainly shop local farmers, maybe eat things in season as well.
Yes.
We can grow our own food as well.
Well, I mean, I live in a condo with a balcony that's 1 by 1 meter. I can't grow anything.
Yeah.
But plus, as somebody, this is a side note, but I was born in the Soviet Union, and we used to get plots of land to grow our own food because there wasn't always food in the stores. And that's not really a practical solution.
Yeah. I meant, so, you have a few, you can grow some food in the garden because it makes you appreciate, I suppose, pollinators.
It does. And if you enjoy doing it, that's great, because that tends to be actually a lot more delicious because they're fresh, they're in season. Berries that you're going to grow, like raspberries you're going to grow in yourself, will never compare to the store-bought ones. Tomatoes you can grow in your own backyard will never compare to the store-bought ones. I can assure you of that.
Yeah.
It's kind of a novelty. It's fun. It's great for people who enjoy it. It's not practical, but maybe we can support the people who are doing it commercially so that it becomes practical for them.
Yeah. Good point. Okay. Well, thank you so much again. And with that, I guess we'll leave it to our next. We have something coming up in March. David, I think you're on at March. He's talking about what he's done in Calgary. I think it's on March 9. Actually, I don't have the date with me, so I apologize for not knowing that in advance. I should know these things. It's quite an interesting story too in Calgary. David did this Pollinator Way. He works for the city of Calgary in transportation, and decided, let's have a corridor where we're going to plant all these pollinator-friendly plants, native plants. And in fact, he had researchers, Lincoln Best is a researcher actually on our board as well. And I don't know if he was the one who discovered, but a whole team was out there trying to examine what kind of bees, what kind of pollinators were there, and they found a rare species of bee, a cuckoo bee, I think. And so yeah, they almost extinct actually. So if we plant it, they'll come.
Yeah.
So lots of thank yous from our audience, and a lot of gratitude. Thank you so much for your generous time that you've.
Thank you so much for having me, and thank you for listening and being interested.
Oh yeah. Thank you again. All right. Have a good day, everyone.
Thanks.
Bye-bye.